Do I really need a microphone? Do I need a microphone, John?
I doubt it, but
I know, but you know something? Who has the last word? Right.
first and last.
It's a joke. This is a joke we have, right? It's my great pleasure today to introduce John Maris, a professor of pediatrics at the Perelman School of Medicine, who's going to speak about a genetic basis of cancer. Oh, right. It should be a genetic basis, not the, because there are probably several genetic bases for cancer.
Fair.
John received his bachelor's degree at a little-known place called Wheeling College, but then he saw the light, and he came to Penn, and he's been here ever since. So his MD was at Penn, and his training was at Penn in pediatrics and in pediatric hematology, oncology, but mostly oncology. And he rose through the ranks from assistant professor to professor with tenure in 2010. The most amazing thing about John was that when he was a medical student, he was the first author of a New England Journal paper, which is an amazing paper, because he worked with Audrey Evans and Britton Chance at their laboratory at the medical school, and wrote that paper, and that was the beginning of his illustrious career. Of course, he's received many awards, including being a member of AOA and a member of the Society of Pediatric Research. The things that I appreciate were the Penn Awards, including the Osler Award, which I'm happy to say we share, and the Berwick Award, and also from the Society of Pediatric Hematology Oncology, the very prestigious Oskey Award. Frank Oskey was a professor here many years ago, too. So there's a lot of Penn connections. And John is really going to enlighten us because he knows what he's about. And his work in pediatrics is now not just in pediatrics, but is become important in adult medicine and especially in adult cancer. So, John.
Well, thank you very much, Anna. Never quite know what to expect when you're introducing me, but thank you. I actually have she stole a little bit of my thunder. I have a picture of it. So we'll get to that. I want to thank Anna and Sue for inviting me here. We've been trying to set this up for a while, and from the time that Anna first reached out to me to do this, I turned 55 and got a letter from Penn saying that I'm now a member of this august group. So,
Congratulations.
Thank you. So I thought that genetic basis of cancer, A or the, would be something interesting to talk about. And I subtitled this Relearning Past Lessons because we thought we knew certain things many years ago, and we are relearning things, and as our technology gets a little bit better. And I do think we have some new insights. And as Anna alluded to, a theme of my career, and learned this a lot from her husband, is that children with cancer can teach us a lot about the problem of cancer in general, and that will be one of the major themes that we go through here today. So this is a very abbreviated history of cancer predisposition. It was many years ago that Broca actually wrote a very interesting paper about his wife and her family, observing that many of them had breast cancer, and this was the first known, at least known to me, description of familial cancer. It wasn't until many decades later that Bovary had his seminal paper that proposed that cancer can be inherited through something he called genetic traits, well before we knew what that really meant. And that is an often cited paper as, the first description of genetic predisposition to cancer. But it was Al Knudsen, there's a long time in between here. It was Al Knudsen's seminal paper in 1971 that proposed the first, modern model of why people develop cancer in the first place. And I'll come back to that in a minute because it really, for me, was always the paper that sort of set me in my direction for how I wanted to try to do something impactful in my laboratory, and I think had a huge influence on the field in general. And as I'll show in the next few slides, after Al proposed a genetic basis of cancer and thought that it would take 2 mutational events to get the ball rolling, there was some time before the first cancer predisposition gene was discovered. And Al's, as I'll talk about it a little bit more, his paper was on this disease, retinoblastoma, a very rare eye cancer of childhood. And it was Webb Cavaney and Anna Meadows, our own Anna Meadows, who made some seminal clinical observations about children who had retinoblastoma and other constitutional abnormalities, and finding that they had a break in chromosome 13 that led them to start looking on chromosome 13 where this gene resides. And then, there were many other cancers, and the reason I sort of put this way in 1990, 1991 is that these are all childhood cancers, and it wasn't until 94 where the first adult cancer predisposition gene was definitively localized by Mary Claire King. And so when I was in medical school, Peter Knoll roamed these campuses, and we all knew, thanks to him and many others, that cancer was a disease of the genome. But what I was taught was that most cancers are caused by environmental factors, and it's clearly true that there is a gene and environment interaction. But because hereditary cancer is relatively rare, these families are relatively rare, we were all taught that it was the environment that was the main culprit in cancer. And this isn't a right or wrong, but I think I was led to believe that the genetic basis of cancer initiation, as opposed to progression, was more chance and more environmental than anything else. And the other thing that I was taught was that history is a critical important of being a doctor. Doing a family history is a critical important part of being a doctor. And that if in the field of cancer, if you did a family history and it was negative, then it's very, very unlikely that there's a genetic cause. And that's something that we were all taught, and I think we believed. And I'll show you some data a little bit later that this is probably not the way we should be thinking about the genetics of cancer. So this is Al and his seminal 2-hit curves, and Al's prediction was that if there is a mutation in the germline, then it was much, much more likely to get a second mutation. Al did not predict tumor suppressor genes. He felt that there needed to be 2 genetic events. But this led the whole concept to our understanding of that most cancer predisposition is due to loss of function. Al's model didn't predict 2 alleles of the same gene, but it followed very quickly, and that proved to be true. So again, this paper is, I think, and if you look at, Nature does a little timeline of the most important papers in the history of medicine, and this is clearly one of the ones that is highlighted each time they do that. So Al did his work in the 70s with Louise Strong at MD Anderson, and then there was a period of time where people were searching for cancer predisposition genes. And you can see that over the years, many of these cancer predisposition genes have been discovered. So this is the retinoblastoma gene back in 1983. And then these are all childhood diseases. In the early days, it was WT1 for Wilms tumor, P53 for this rare Li-Fraumeni syndrome, which is an excess of sarcomas, breast cancer in later life, rhabdomyosarcomas. These were all cloned in the early, late 80s and early 90s. And then as technology changed and we had different ways to study our heritable genome, more and more discoveries were made. And so this was a pretty dizzying pace, and today we know of 114 different cancer predisposition genes. They're sort of scattered across the genome, and it's not important to go through any detail here. The only point I wanted to make with this slide, because it will come relevant to neuroblastoma, which we're going to talk about in a little bit more detail. In most of these are colored black because they followed Al's hypothesis that there would be a deleterious mutation in one allele that was inactivating, and then you lose a second allele. So these are tumor suppressor genes. There are a handful of these that are in red where it's the opposite, where the mutation that is inherited is a gain-of-function mutation, actually makes the gene work in a more potent fashion. And we're going to come back to one of these called ALK here on chromosome 2 in a little bit. Okay, so that's a little bit of a background about cancer predisposition in general. And as Anna mentioned, I've spent a large part of my career studying this relatively rare disease, neuroblastoma. And I just thought I'd spend one slide reminding people in the audience what this disease is and why we, and in the next slide, why we consider it still a very important problem to address. So it's a developmental cancer. I was always interested in developmental biology and the fact that this is the genes that we have subsequently discovered that I'll talk about all have something to do with how you put together a normal nervous system. And this is not a brain cancer. This is a cancer of your autonomic nervous system, your fight or flight response nerves. And so these tumors can occur anywhere in the neck, chest, or abdomen where you have a sympathetic ganglion. And it's a true childhood cancer. It's the most common cancer that's diagnosed in utero, and it's the most common cancer of infancy. It is, and as I'll show on the next slide, still an important pediatric problem in that a large proportion of childhood cancer mortality is attributable to this disease. And these two children both look sick, and this very diverse clinical course is something that, as I'll show in another slide or two, Britt Chance and Audrey Evans asked me to try to address when I wasn't really even sure what the New England Journal of Medicine was, to be quite honest. And this is a baby who has a metastatic, so stage 4 metastatic type of disease, but has a pattern that Audrey Evans and Dan DiAngio recognized back in the 70s, where the pattern of dissemination was such that you could predict with a high degree of certainty that this would all go away on its own. So neuroblastoma has, of all human cancer, the highest proportion of cases that will spontaneously and completely remit without any chemotherapy. And so this baby is now, or this young man is now in his 30s and never had any chemotherapy or radiation therapy. This child, and those of us who do this for a living, could recognize the pattern being differently. Neuroblastoma has this we don't understand the reason for this, but a predilection to metastasize to the bony orbit. And the pattern of metastasis here is different, and as I learned later in my career, the genetics of these tumors are very different, and this is a relentlessly progressive disease. And it's out of, I'm sorry, I got the order out a little bit. And the reason why, so this is when I got into the field, and most patients with high-risk neuroblastoma would pass away. Survival rates were in the 30s or so. And you can see we've made progress. But if you look at these curves close enough, we're really just pushing the curves out, right? And, overall survival is only modestly improved. And how have we done this? Well, we've done this by a dramatic and we've tried to quantify this, it's at least a 3 or 4-fold increase in the intensity of the therapy they're receiving. So we've gotten better at giving chemotherapy safely. We have procedures like myeloablative chemotherapy and stem cell rescue. We use a lot of radiation therapy. And so one of the important lessons I learned very early on, and Anna was critical in my understanding that it's not all about survival curves. This was the first neuroblastoma patient I had when I was a fellow at CHOP, Bobby Burns. And he is now a long-term survivor, so he's out here. But he wasn't destined to be this short. He has severe growth hormone deficiency. When I saw him this day, his triglycerides were in like the 6,000 range. His cholesterol was some sky-high number because he hadn't been seen in the survivorship clinic for many years. He has cardiomyopathy. He has not had a second malignancy, which is something that happens in about 15% of neuroblastoma survivors when you follow them out long enough. So he has not yet had a cancer. He actually became a jockey and was actually a competitive jockey. So he's really learned how to adapt. But he is emblematic, I think, of the whole concept that cure is not enough, and that we have to be thinking all the time about modifying our strategies so it's a quality outcome. So I wanted to show this slide, and Anna mentioned it. I get asked the question all the time, how did you get interested in neuroblastoma? And Anna already sort of mentioned the story that I came and was very lucky enough to have a technician job in Britt Chance's lab right when I got out of college and was trying to decide whether to go to medical school or graduate school. And literally the very first day of work, he introduced me to Audrey and said, there's this young pediatric oncologist who wants to try to figure out why neuroblastomas regress. And so this is Dan DeAngio, who was also part of this collaboration. And so this was one of several projects that he asked me to participate in while we were trying to measure the energy, ATP quantity, phosphocreatine, phosphomonoesters, and try to predict why some neuroblastomas would progress and regress. And we actually did figure out that we could at least correlate these patterns of phosphometabolites with progression or regression. Did not change anything in how we care for the patients. This is a historical artifact at this point, not used clinically. But it did really, I did get a chance at a very formative age to meet these incredible babies and their families and dedicated physicians, and it sort of just hooked me into wanting to try to tackle this problem and to use this disease as a microcosm of the problem of cancer in general. So back to cancer predisposition. So Al's paper on retinoblastoma is one of the most highly cited papers ever written. He wrote another paper a year later that focused on neuroblastoma that is very infrequently cited, but he made the same observation that there were a subset of patients who seemed to be predisposed to develop cancer at a younger age, and he predicted that was because of a genetic predisposition. And one of the things that he wrote in the paper that I read early on and just didn't make sense to me was that he said that about a quarter of neuroblastoma patients would have a hereditary predisposition. And that did not jive with my fellowship training, where I saw a lot of neuroblastoma patients, but very few that had a family history. And so this was something that I didn't understand, and as I'll come back to at the end, I think it was very prescient that he was able to actually think about this at a time where most of us would not have believed that any number this high would be right. So also during my fellowship, and this was a family that Anna took care of, I got to meet this patient who was diagnosed at the time when I was a fellow. So this is clearly, a case where neuroblastoma, all the filled circles. This is a grandmother who had never had clinical neuroblastoma, but she had a large tumor in her posterior mediastinum that was probably a neuroblastoma that spontaneously differentiated. And so this is a family that led me to want to try to figure out what's causing this in the family, and really was my intro into trying to study the genetic basis of neuroblastoma. Now, we spent a lot of time. So this was my fellowship project, and Garrett Brodeur, whose lab I worked in at the time, proposed this to me and said, this is, I think, going to be a relatively straightforward second project, because all lines of evidence suggest that the gene is going to be on chromosome 1, and we should be able to figure it out just like they did with RV1 and WT1. But that turned out not to be the case. And so I started this project back in the early 90s, and we slogged away on this for a very, very long time. And it wasn't until 2008 that we were able to figure out that the vast majority of families that have neuroblastoma have a mutation in this gene called ALK, which is the anaplastic lymphoma kinase gene. And unlike the BRCA1 mutations or the RB mutations, these were all within a part of the protein called the tyrosine kinase domain, which is generally the business portion of about 600 or 700 proteins we have in our cells, that when these become activated or phosphorylated, they generally are signaling something to the cell. And so ALK is a protein that is expressed very early in neurodevelopment. We didn't know this at the time, but it's subsequently shown that it's expressed very early in neurodevelopment. It is activated through normal interactions with ligands. This is something that sits on the cell surface and signals inside of the cell. And then it is turned off sometime during late fetal development when a nerve cell is sat down and differentiated. And these spelling mistakes here that I'm showing in the kinase domain, these all we have subsequently shown, and we'll come back near at the end of the talk if we have time, to make this protein behave in an abnormal fashion in these cells and to activate signaling, and is predisposing to cancer. So that took us some time to figure out the major familial neuroblastoma gene. In that pedigree that I showed you in the a few slides ago, that Anna took care of, they did not have an ALK mutation, and so that was one of the lessons. So they have, that family segregates a mutation in another very important neurodevelopmental gene called FOX2B, POHX2B. The interesting thing about that family and that mutation is that children who have mutations in ALK are, other than neuroblastoma, developmentally normal, as best as we can tell. Children who have mutations in that gene called FOX2B generally have neuroblastoma, but other disorders of the developing nervous system, generally Hirschsprung disease, or something called Ondine's curse, which is central hypoventilation syndrome, where these patients don't breathe. And we've learned a lot about the different types of mutations that cause either just neuroblastoma or these associated conditions. But these ALK mutations are not syndromic, whereas the FOX2B mutations are syndromic, just like they were in the family that Anna took care of. So in the next couple slides, I just want to go through how things have changed. So there's been a lot of studies where you collect the families and then you look for a common gene that is segregating with the cancers, sort of like we did in that 2008 paper. But as technology has advanced and the cost of doing sequencing has plummeted, there are more and more studies where we're gathering an enormous amount of sequencing data from patients with cancer. So the next two studies that I'm going to show, one from University of Michigan and one from St. Jude, were not done with the idea of looking, these were not done for cancer predisposition purposes. So this group here is very interested in, as many of us are, in precision oncology, this idea that you can take a cancer sample, sequence it, find mutations where we have drugs that you can match to, and that you'll have new targeted therapies. A lot of progress in this area, a lot of challenges. Now, I don't expect you to see this heat map at the bottom here, but what they're trying to show you here is that they had about 100 patients, 100 consecutive patients with pediatric cancer at the University of Michigan. They had sequencing done of their germline DNA and their tumor DNA. And the reason the germline DNA was done is that just for technical reasons, it's having the germline makes it much easier to interpret the somatic DNA results because of many different polymorphisms, and so it's done as almost a control. And then these little dots down here are their inference of what is potentially actionable. So if you're a melanoma patient and you have a mutation in a gene called BRAF, there are about 4 or 5 different BRAF inhibitors out there, and that's considered potentially actionable. In this study, they found very few examples of potentially actionable mutations in the tumors of these patients. But what they did find was that about 10% of patients, and I know you can't see this, this is highlighting germline, and so each one of these things are mutations in the germline that they didn't expect. So none of these patients had a family history of cancer, and about 10 to 15% of them had an RB1 mutation, a P53 mutation, some mutation that based upon what I showed you in the very beginning, we now know is a cancer predisposing mutation. Many of these were mutations that predict adult onset cancers, APC mutations, BRCA1, BRCA2 mutations. So that was an important lesson from this study. And the thought is that, and what we're trying to grasp is if a child has a classic BRCA1 mutation, did the cancer that arose in that child have anything to do with that mutation, or is that just important for later in life? And that's, we'll end talking about that, is that the answer to that question remains unknown. So the St. Jude paper, and I again apologize if this is just, so they had over 1,000 patients in their study, a variety of different childhood cancers, and this is just showing that what we knew, P53 was the most commonly mutated gene, but all of this data here is just quantifying the fact that, again, in their study, exactly like the Michigan study, about 1 in 10 children had a classic cancer predisposition mutation. So that was a big surprise. Two other points I thought were really important from this study is what I mentioned in the beginning about the family history. So if you gathered up the 100 or so patients they had with a germline mutation, classic germline mutation, only 40 of those subjects had a positive family history. And I can say this now because I reviewed this paper. At first, they didn't have this in there, and we actually had them go back and do chart reviews because they have that ability at St. Jude, and they did a very nice job of going back and doing very good chart reviews, and only 40% had a family history. But conversely, of the 900 patients who did not have a germline mutation, 42% of them had a family history. So this just goes to show that, yes, I'm not trying to say that taking a family history is not important, but a lack of a family history does not mean that there might be something important in the genome of the patient that you're seeing. And then the other point here, and I'm not going to go through the exact data, but these bar graphs are showing the proportion of so a single base is mutated in any of these cases. And in a typical situation, when you think of your classic Mendelian inheritance, it's heterozygous, so about 50% of it's mutant. But several times they found cases where a smaller fraction was detected. So this is so-called genetic mosaicism. They did a lot of nice work to prove that this was true and it was not a sequencing artifact. And so that suggests that, that many times these are not inherited, but these are de novo events and likely not in the sperm or the egg, but sometime early in gestation. And the influence of germline mosaicism and how much that impacts cancer susceptibility is still a wide open question. So we've been very active in this area, and a young assistant professor at CHOP has really been leading this work. So they had 1,000 patients in the St. Jude study of a lot of different diseases. We have 868 germline DNA sequences from neuroblastoma patients. I'm going to show at the very end some of what we've learned from that to date, but same sort of lessons. There are mutations in genes that we didn't know they were associated with neuroblastoma. They're not ALK, they're not FOX2B, that seem to be damaging, seem to be causal, and may be important for the individual patients. We have this new, very interesting opportunity that the big problem with all of the studies to date is that in general, we in the pediatric oncology community, when we collect samples from patients, it's just the patients. If you're studying genetics, it's very good to know about inheritance. And, but in general, in CHOP and in the Children's Oncology Group, we don't have samples from the parents. Andy Olshan is an outstanding epidemiologist, and we teamed up a few years ago to collect so-called trio germline samples. So we have a bunch of these. And this will, for the first time, allow us to quantify what proportion of the germline predisposition is de novo arising likely in the sperm or much more frequently in the sperm than in the egg of the father or the mother, or how many of these are inherited. I just wanted to mention as a passing this, probably none of you have, or very few of you have heard of this Gabriella Miller grant mechanism. But we found it very interesting and just thought I'd mention it. So Gabriella Miller was a girl who had a brain tumor. Her parents were lobbyists, and they somehow figured out that you probably all remember a time when you filled out your tax form and you could check a box and $3 would go to one of the candidates. And you probably recognize you don't see that anymore, that it went away about 3 years ago. What Gabby Miller's parents figured out was that money was going into some black hole and was not being spent. And it never went to either party and was sitting there, and there was a little over $30 million just sitting there. And so they got an act of Congress to appropriate that to the study of childhood cancer genetics. And it got political, and it broadened a little bit, so it's cancer genetics and cancer and structural birth defects. So this is a funding mechanism that looks for these sorts of sample sets to try to understand more about the genetics of the disease, because this was something Gabby's family was very interested in, is why did she develop brain cancer in the first place? And then everything that we've talked about to date in this lecture is been focusing on mutations in the coding region of a gene that makes a protein. And I'm probably not going to get into this for sake of time, but it's very, very clear now that many of these mutations that are important in cancer predisposition are outside of the protein coding genome, but are in regions that dramatically regulate expression of genes, so-called enhancer elements. And this is a very big area of research in the cancer predisposition field, is how do these non-coding mutations influence cancer predisposition. So I want to talk briefly about common polymorphisms. So this is from Terry Manillo a few years ago, now many years ago, where it's a very nice model of the genetic basis of all disease susceptibility, not just cancer, that you have we all have in our genome spelling changes, polymorphisms that are very common. Some that, 50-50 chance that you have the polymorphism, and then some that are incredibly rare. And these rare things are what we call mutations. And then each of these have an effect size. So common polymorphisms are often recently evolved and have a very subtle influence on any sort of phenotype, depending upon what the gene does. Whereas these rare mutations, as we've been talking about, have a very big effect size. So if you're born with a mutation in one allele of the retinoblastoma gene, it's over 90, probably 95% chance that you'll develop the disease. So it's a very high effect size. If you have a polymorphism in a sugar metabolizing gene, you have maybe a twofold increase of developing diabetes. And so everything that we've talked about to date is up in this area, but clearly this is this influences dramatically cancer susceptibility. So you get at this by doing something called a genome-wide association study. This is GWA here. And what this slide is summarizing is about a decade of work, myself, Sharon Diskin, Yael Mosé, to do a very large case control series. So in a disease that only afflicts about 600 patients in the US each year, it's taken some time to get these sorts of numbers, but we've now genotyped or studied polymorphisms across the genome in a large number of neuroblastoma cases and an even larger number of controls, thanks to our Center for Applied Genomics at CHOP. And we're nearing our goals, and we'll probably finish the genotyping in another year or so. And this picture here is showing each of these dots is a polymorphism. So a known variation in our genome that helps make us look different, have different color hair, et cetera, et cetera. And anytime you see this stacking up, this is on the y-axis is this statistical significance. And so anything you see something stacking up, there's an association. So what that means is that there is neuroblastoma patients, in the case of this LMO1, are much, much more likely to have a particular risk allele than children without neuroblastoma. And so these are all different polymorphisms that are highly statistically significantly associated with neuroblastoma, and a big focus is how and why. There are, I think, two important lessons that I'd like to just leave with for this discussion is that I said back in the beginning that there are different neuroblastoma phenotypes. There's the high-risk form of the disease that is relentlessly progressive, and then there's the lower-risk form of the disease that can spontaneously remit. Many of these polymorphisms that we've discovered predict for one or the other. And what that says to me is that these diseases that look the same under the microscope and we call neuroblastoma, and then we label them low or high risk, are actually very fundamentally different at a genetic basis. So what gets the ball rolling is very fundamentally different. And it's not the way we think of colon cancer, where there's an accumulation of somatic events, that these are hardwired to be benign or malignant. The other important lesson here is that many of these genes are critically important in normal neurodevelopment, and after the process of cancer initiation, the cancer cells figure out mechanisms to either stochastically or epigenetically select for them. So in other words, and I have a slide, I'm going to probably just go through it, skip over it first time, but just to tell you the punchline, this gene LMO1, there's a polymorphism in it. We figured out which one it is. It's much more likely to be present in neuroblastoma patients. In the patients who have this polymorphism, once the tumor arises, this protein, which is a transcriptional cofactor, gets dramatically overexpressed, and the cells become reliant on it. So that if you can when we genetically ablate LMO1 in established neuroblastomas, the cells die. And so a big part of our effort now is, are there ways to drug these things and take advantage of a fundamental defect that arose in the germline of the patient, and I'll come back to that point near the end. So I'm just going to skip through this very quickly. That's what I just said about LMO1, and then I just think that this is interesting. I just wanted to share it, that I have a new collaboration where we take our genome-wide association signals, and in order to prove to ourselves that they're important in tumor formation, we have a way to pretty rapidly model these in the zebrafish. And this does what they're supposed to do, or at least what we think they should do. So this is a picture of an eye of a fish, and as I mentioned in the beginning, neuroblastomas in kids, the high-risk form metastasized to the bony orbit, and that's what this is here in this fish, where the tumor is in the orbit. So we think that these are good models, and we're using these fish. These are small fish. You can do screens by putting drugs in the water and other things, so this is an important model for us. All right, so I want to come back to ALK to try to drive home some of the reasons why we think that this is clinically significant. A lot of the work I'm going to go through now is from Dr. Yael Mosse's lab. But when we first started looking for this, our dream was that any gene that was important in a family will definitely be important in terms of genetic counseling and screening, but if it could become a therapeutic target, that would be an even more important outcome. And what this Venn diagram is showing is that we have about 114 known cancer predisposition genes. Then we have almost 500 genes that we know are important in established cancers. And there's an overlap. So there are 49 genes. This one ALK that I'm going to talk about is one of those, where it's important in the initiation of the cancer, but also can be mutated without that initiating event. And I'll tell you what I mean about that. And with this, for those in the cancer field, we've seen this probably too much. This is a famous review by Doug Hanahan and Bob Weinberg, where they sort of laid out the hallmarks of cancer. What has to happen to a normal mammalian cell for it to become a cancer cell? And I only show this because our cancer predisposition genes fall into all of these categories. So this model is important because these are thought to be Achilles' heels that one can think about when thinking about how to treat or how to drug cancer. So this is a very busy slide. I only want to make one point. So after the discovery of ALK mutations in the germline, we very quickly wanted to look at tumor cells. And so these are 222 neuroblastoma patients lined up in columns. And these are things about their gender, their age, and these are chromosomal changes. But then these are the genes that were mutated. So this was a sequencing project we did. And a very important point is that a lot of patients had no known somatic mutations. So pediatric cancers in general, at the time of diagnosis, have fewer somatic mutations than adult malignancies, which we think makes sense because of the less of an environmental influence. But the gene that had the most frequent mutations was this gene called ALK, and then Yael went out and quantified it. And so high-risk subjects, about 14% of them have a somatic mutation. So the germline is normal, but they have a somatic mutation. I'm going to go through this relatively quickly, and there's data, but I'm going to just try to tell you the take-home point. What this slide is trying to emphasize is that there are about a dozen mutations that have been identified to activate the ALK kinase. And in this field of trying to understand how to take advantage of this therapeutically, it's not a binary problem of ALK's either mutated or it's not. And so what this slide is trying to show is that there are some mutations that so potently activate the kinase that all of the known drugs that we have, until one that came out recently, could not get the ATP out of the pocket. So these are kinases. They, when these mutations are present, they bind ATP, and and they tend to hold on to it with high affinity. Most of the small molecules that we have to try to interrupt kinases displace that ATP from the binding pocket. And so these mutations, we can displace the ATP, but in these mutations it's much, much more difficult. And these are data just quantifying that. So when we go to the clinic, and the trial's already been done, there are patients who will respond to the kinase inhibitors, but there are many who won't because there's just no chance that we can displace the ATP. So what Yael did because of perseverance is some pretty straight, this is I think a very straightforward but very, very important experiment. You don't cure cancer with a single drug. The idea of all this is we're not going to just come in with an ALK inhibitor for these patients and get rid of everything else. If you're really going to make a difference, you need to figure out a way to move it into the upfront setting and integrate it with existing therapy. Hopefully, over time, we'll begin to be able to dial back some of the toxicity of the therapy. But two chemotherapy drugs that we commonly use, we always use nowadays, to initiate therapy for children with neuroblastomas are topotecan and cyclophosphamide, two standard cytotoxic agents. And so these are all patient-derived xenograft models. So, well, this one's not, but in general, what we do is we try to use systems where a patient's in the clinic, the tumor is removed, it goes directly into a mouse so that it never touches plastic, which can change a lot of things about a cancer cell. And so this is from a patient I took care of myself, Felix, and the parents wanted to name this after him. And what each of these are showing is that the tumors grow in mice when they get placebo. If you get this drug crizotinib, which is the first-line ALK inhibitor, they respond, but then they always regrow. So they're continuing to be treated throughout the entire timeline here. If they get topotecan cyclophosphamide, they got it at week 0, then again at week 3, then again at week 6. It responds, but then they break through. And so this is what we see in the clinic all the time. But you can't even see it in green here is the combination of these three where they get deep and sustained regressions. And so you do that in both the sensitive mutations and in the resistant mutations. And the reason why I think that this paper is so important is that it's the preclinical justification to this overly complicated clinical trial, which for the purpose of this audience is that children with newly diagnosed neuroblastoma will get their tumor sequenced right at diagnosis, and if there's an ALK mutation, then they'll automatically get the standard chemotherapy with this drug crizotinib, which is the ALK inhibitor. And we think that this is going to be a critically important study, very similar to how the drug imatinib got integrated into frontline ALL care for children with Philadelphia chromosome positive ALL. And Steve played a very important role in that story. You know, technology has been amazing, and we now have the ability to really dissect the heterogeneity of these cancers we're dealing with, and it's stunning how complex these cancers are. We now have the ability, so when I started sequencing, you know, we could maybe detect something that was present in about 30% of the population. Now we can detect things reliably that are in 0.0001% of the population by sequencing very, very deeply. And this is just a subset of data to show that if you just look at this gene ALK, and these are all different tumors, whereas with standard technology, about 15% of them have an ALK mutation. If you go this very deep sequencing, it's more like 40%. And so these are the clonal ALK mutations, near 50%, that we typically detect with standard technology. And these have all been validated. These are very low fractions, but they're there. And in data that I'm not showing you, what we've been able to show that if you look at paired diagnostic and then relapse samples after standard therapy, very frequently these emerge as clonal, suggesting that these subclonal fractions are dictating chemotherapy-resistant relapse. And so the point of this slide is that Well, 15% is a small or large number depending on how you think about these things. We think that there's going to be a much larger fraction of neuroblastoma patients who will benefit from ALK inhibition therapy, and there are many, many other genes that have mutations in the subfraction level that we do have targeted therapies for. So we think that this could be importantly clinically relevant. Skip over that. So, last couple of slides. I showed this in the beginning. Terry Manillo's, these are the different DNA variation, these are the effect size. And through all of our sequencing efforts, we now have a pretty long list of genes, other than ALK and FOX2B, that have some sort of influence on neuroblastoma development. I can tell you that these are in the germline of several patients with neuroblastoma. Some of them make sense, others are surprises, and it's going to be another significant amount of work to understand the mechanisms and the potency by which these influence neuroblastoma susceptibility. On the other end of the spectrum, we've discovered a lot of these very common alleles, and we talked a little bit about their influence. And then where we are now is that there's this whole area in the middle of relatively rare but higher potency mutations. Sharon Diskin has a beautiful story coming out where this copy number variation, so on the short arm of chromosome 16, there is a reproducible chunk of DNA that's missing in a small number, in about 1% of neuroblastoma patients. This is seen in 0.0001% of non-neuroblastoma patients. But these are the same copy number variation that has been associated with autism, and we all know that there are a subset of neuroblastoma patients who are autistic, and we think that there's going to be a lot of these sort of aberrations that explain some of the complex phenotypes that we see in our patients. So I'm going to skip that. So Al was right. I mean, I do think that he was present in putting out there that the genetic variation explains a lot more of pediatric cancer for sure, and I think cancer in general, susceptibility. But I would say, and I had a lot of great discussions with Al up until the time of his death earlier, or late last year, and he was humbled as well about how complicated it's gotten, and I don't think that anyone in the 70s could have predicted all these gene-gene interactions and the different effect sizes. And there's a hell of a lot to do in terms of understanding the ecosystem of how these work, how they interact, and so I think that many of us in this field are going to be busy for quite some time. I have a rather large lab, and I want to thank everyone. They've all contributed to this. We get all of our samples through something called the Children's Oncology Group, which has been critically important. And then I mentioned Yael and Sharon a few times through the talk. They've really both trained in our fellowship program and have both developed independent laboratory programs that are outstanding in their own right. And I'm got their permission to share a lot of their data with you today. So thank you for your attention.
So I think we're all excited about what looking for genes can help us therapeutically. What's the lesson in this for patients with pancreatic cancer, which, for example, takes more lives than neuroblastoma in 20 years?
So, all pancreatic cancers have RAS. There's a whole NIH, NCI section on RAS to try to get at that. So there is a vulnerability there. We just have not been smart enough to get at it. And if we can ever learn how to drug RAS, it's going to help pancreatic cancer patients, a large number of lung cancer patients, and a fair number of pediatric cancer patients. I think that knowledge is power, and there have been some unexpected findings. Many pancreatic cancers have a hypermutant phenotype. So if, as a cancer evolves, they develop defects in DNA damage repair genes, they can end up with mutation frequencies. So I was showing you frequencies of about 15 to 30 mutations per megabase. They can end up with 1,000 or 10,000 per megabase, this stunning number. And what's become very, very apparent recently in our own work and others is that those are the cancers that are responding to checkpoint blockade therapy.
So there is a value in doing deep genetic explorations of all.
I think that the risk in the field right now is that there was such a rush to get the sequencing done and get some papers published that people have looked at the tip of the iceberg and moved away from the data. And I think that these sorts of things have come from additional sequencing studies as well as going back and mining existing data. So yeah, I do believe.
Speaking about the pancreatic cancer, I think group in Johns Hopkins about 2 months ago, they published a paper about relating the pancreatic cancer to genetic variation, exposition, and mutation, and any idea about really how the pancreatic cancer related to some mutation or genetic disposition?
So, I mean, just like in the pediatric cancers I've talked about, all the adult cancers, when people have looked at germline, they're finding predisposing mutations, or they're finding mutations in genes that were not previously associated with the histology of interest. And so, yes, I think that's going to be relevant. And I think pancreatic cancer is probably one of the best examples of the heterogeneity of the ecosystem. Especially pancreatic cancers get such an inflammatory component. But there's mutations in that part of the pancreatic cancer as well as the bona fide cancer cells. And so understanding not just the yes-no is a mutation there, but in what compartment is it and what frequency, I think, is another really important area. Okay. Thank you all very much.